one.
Okay, good evening. This is Dr. Ken Shen from West Virginia University. And today I will talk about something genetics. And you already know probably, which is DNA, a little bit of DNA structure. And hopefully we have time to go into the DNA replication. So when we talk about the DNA, we need to talk a little bit about history. So here is the thing. We need to talk about is back in 1920s to 1940s. There is several very important research experiments to make the progress. People find the DNA and its structure. So the first thing what I want to mention is that people try to solve the problem called transformative principles. Now, who is the first person to solve the problem? Is F. Griffiths. He's a British biochemist. The very famous study in 1928, he did for Streptococcus pneumoniae in mice, and to see whether the mice has died or still survive. Now, we want to mention something. For Griffiths, by the time in 1928, he's not trying to really do some genetic stuff. Because at the time, is 1980s, we had another pandemic called the Spanish flu. The Spanish flu kills millions of people in the world because there's no vaccines, there's no antibiotics. So at the time, they're trying to solve the problem to develop a vaccine. Therefore, he started to do some of the research. So first of all, what is Streptococcus pneumoniae? This is a gram-positive diprococcal. It's a lancet shape. And even today, this is still responsible for 80% of CAP. We call it community acquired ammonia. Now, what's the symptoms? You know about that is chest pain, cough, and fever. Now, right now we know we can use antibiotics. We also can using a vaccine called the PVC vaccine. The PVC vaccine can do both on the kids, adults, and older people. Can solve the problem. But back in 1928, there's no way to do. So he tried to do some of the research. What's the research he did? For Streptococcus pneumoniae, there is a two different shape. Number one, we call it smooth one. Smooth one has a heavy capsule, which is a shining, okay, shining colors, that's a capsule. The capsule is important for anti-phagosome. Now he had another one, which is a surrogate, a rough one. Now what's the difference between that? The smooth one, which is virulent, what means? The mice will be died. What means the rough one? Is non-virulent, which means the mice will be survived. So he did a bunch of research. What he did? Okay, let's say we had a smooth one. We're gonna go into a mice. Okay, let's draw a mice, very simple. The mice is dead. Second, he had a rough one. The rough one gonna be injected a mice. The mice is survive, is alive there. Okay, what are you gonna do next? I had a smooth one. However, I will heat treat it. Okay, they use an autoclave, old autoclave methods. Then they go to inject a mice. What's gonna happen for the mice? The mice gonna still survive because the bacteria is dead. Okay, what he did is another one. He using a rough one and then combined with a smooth one. And the smooth one also heat injured, or we say heat treated. And then they combine them together and then go to a mice. Now what happened? The mice is dead. So what this tells us? This tells us there is something happened. Even this smooth Streptococcus pneumoniae has been heat injured or heat treated. There is something going to transfer here, which is some genetic information. And also very important, from this mice, he isolated smooth Streptococcus pneumoniae. That is important. Okay, this is follow the Koch postulate, which means a pathogen infectious live animal, they will get the same disease, and the colony can be re-isolated. Okay, this tells us there's some genetic information. They don't know whether it's DNA or RNA, or like protein even. They don't know what it is. So they finished the research. They even know this can be transferred. And then we call it a transformation. Now, Griffiths did this research in 1928. His lab is continued to do, working very busy day by day. They have lots of mice, okay? Hundreds, millions of mice, thousands of mice sacrificed every day, every year. So they have to blend them, to isolate it. They want to find what type of the material is genetic information. Because his research is so focused on the mice, so focused on the animals, it's lots of work. So there's not much progress in 1930s. Not much progress. And the reports, the history report says he made some progress in 1940 to 1941. But unfortunately, he was died in the lab because of the German bombing during Second World War. Very unfortunate, very talented people is killed by the German bombing during Second World War. Okay. However, people have to continue to do the research about that, to find this genetic information. It's Second World War. Everybody is busy. There is a group of folks in 1944 in Rockefeller University. Right now is Rockefeller Institution of Medical Center in New York City. A group of people is Avery, McCarty, and McLeod, or we say McLeod. They did a serious research, which in a bacterial medium, agar medium, to verify this genetic information is DNA, and the DNA can be transferred between bacterial cells using transformation. Okay, how they did that? They had the first tube. Okay, this tube is we call all Streptococcus pneumoniae. I'll just say all cells. So you have a second tube. Is S ammonium which is extract. So they're going to combine them. Then they're going to do what? Centrifuge. Now during the centrifuge, they also add some of the antibody. And this antibody will prevent the agglutination of rough streptococcus pneumoniae cells. And then they do another job, which they're going to be plated onto the agar plates to get a single colony. Okay, the first thing they did do this. They get a very beautiful single colony, which means there is a DNA, and they transformed. Now they want to verify it is DNA, it is not RNA, and the others, what they do. Okay, then they do a series of the sets. Okay, sets, we had the 2 tubes. Okay, this is R cell, this is S cell. Same thing. Then same thing, they're gonna add DNase. When you see ASC, it's an enzyme. So which means they're going to hydrolyze DNA. Okay, they did on this agar plates what they find. There is no colony, no colonies, no transformed. Okay, then they think it's maybe DNA, but we need to make sure it is not RNA, is that right? So what we do, they prepare another 2 tubes. One is R cell, another one is S cell, and they're going to add RNase. When they add RNase, then they go to here, what they find? They still find a single colony there, which means this is DNA. It is transformed. Okay, we need to make sure it is DNA. We need to make sure it is not protein. So they have another set. Still the tube is rough bacteria. And we have a smooth one. And then we're going to add protease. Once you add a protease, you go here. What you find? Still the same thing. You see the DNA is there, the bacterial colony, which means the DNA is there, and it's transformed. So, every McCarty and McLean, they are from the culture media standpoint, they verify this genetic information is DNA. And they are going through transformation methods to transfer between different bacterial cells. This is a little bit advanced compared to this research. Why? It's done independently without animals. They do not do anything, rely on the animals. Okay? So once this research done, then the people gonna think about, we're going to do something else. We want to verify, continue to verify it is DNA. These are genetic information. So how they do? This is we're gonna go to is 9052. You know there's something happens in 1953. We're getting there. 1952. A very famous research. Hershey and Chase experiment. This is not candy bar, not chocolate. Okay, Alfred Hershey. This is not Chase Bank. This is Master Chase. Alfred Hershey is still there. He's later is a professor in University of Oregon at Eugene, Oregon. Back in 1952, they both are research associates, and they are doing a very important research from another standpoint to verify that's DNA, the genetic information is DNA. How they did? So we had a bacteria. That's not a problem. So bacteria has a genome. Okay, bacteria has a genome like this. Now, they're not going to use animals, not going to use culture media. What they do? They're going to use a bacteriophage, which is a virus. This looks like that. Okay, what is a bacteriophage? A bacteriophage, they could eat a bacteria. So they could eating bacteria. Phage means eating. Now what is the structure? The structure same as other virus. It's very simple. What's the top? Protein. Or we say protein coat. Capsid or capsids. What is this inside guy here? This is genetic material that could be DNA or RNA, never both. Remember. And this phage they use is a T2 phage. The T2 phage is a temperate phage. The temperate phage, which means they're not going to cause the burst of the bacterial cell. They will be integrated their genome into a bacterial cell. And later on will survive there. So we call it a temperate phage, and that's a T2 phage. Now some of the bacterial cell, a bacterial phage could cause lytic. And we mentioned later on. Okay, so what happened here? We need to verify the genetic information is DNA, so we're going to have to label something. How are they going to label? We're using some chemicals. The chemicals is isotope. Some radiation they are put. Okay, the outside is the coated protein. We're going to label them S35. Now inside is genetics. We make sure it's RNA, DNA, or maybe DNA. We're going to label them P32. Now why we label those two? Sulfide is always in the protein. It's not going to be in the DNA. Phosphate is a major component of DNA. We're going to get to that real quick. It's not going to be in the protein. So that's why they label it. Okay, after they label, they use isotope to label them. What kind of results looks like? The results will have several things. Number one, you know the phage, when they go into a bacterial cell, it's an obligate parasites. So they will be using the bacterial cell machinery to synthesize its own. So we find both P32 and S35 in the new phage. It doesn't mean anything. Now some of the bacteria we didn't find anything because it's not integrated successfully. We didn't see anything. However, we do see a lot of the bacteria. What happened? Their DNA has been integrated with the genome from the bacterial phage, and it shows P32. What that means, which means this is the DNA from a bacterial phage is successfully transferred from a bacterial phage to a bacteria. This is later on we know it is called transduction. So remember, three things we can transfer genetic information between bacterial cell to bacterial cell. What are those three? Transformation, transduction, and conjugation. This is rely on F pili. Okay, we don't mention this one here. Now, the story is, when they mix bacteria with this phage, at that time, it's difficult to do. They don't have like a huge centrifuging machine like right now they have. They cannot isolate the DNA since they are doing the research for DNA. What they do? They blend it. They finally find the kitchen blender is a good way to separate the bacteria and a virus. Okay, when they blended, what they find? They find all those virus, bacteriophage, is on the top. And they find all the bacteria, they are actually is a precipitation at the bottom. So, using a kitchen blender, they blended, they successfully isolated to differentiate between bacteriophage and the bacteria. That's why they can do this research. Now, the question is there going to be 100% to separate them? Of course not. The report says there is about 1% is mixed. Even in a bacteria, it's mixed of they find P32 and S35. But it's not going to bother the results. The results still tells us that genetic information is DNA and it's transducted. Now we go back and want to mention why they want to try to do this research. This is 1952. You know at the time, antibiotics is there. The vaccine is starting to develop but not widely used. There is some idea, people say, if we want to combat, to conquer the bacterial disease, can we drink virus? Some folks said, okay, let's try it. So they did do something. Some people tried drinking virus. Of course, they failed. It's not successful. But you know today, there is a bacteriophage therapy for certain disease in the veterinary science area, not using the human being. But I just want to let you know, back in 1950, they did some research. The original idea is trying to see the bacteriophage therapy for the human being. Okay? So with these three important research, we almost get done for the DNA. Now what is DNA then? We're going to talk about the real sciences for the science. Okay, what is DNA? Can you tell me the full name? I ask my students, maybe 60% of them don't know it. What is DNA? 2' deoxyribose nucleic acids. Nucleic acids. Okay, now what is 2' deoxyribose? We're going to go back to review a little bit of chemistry. Okay, 1', 2', 3', 4', and go here, 5'. We have OH, H, H, OH, H, OH. Here, this O is gone. Okay? That's why we call it a deoxyribose. So this O is gone. Okay, what the deoxyribose they connected? Right here, we're gonna add a base. What are those bases? Can you remember it? Do you still remember it? You should know. You learned it in your biology or in your high school. What are they? The base could be two different morphology. It could be a 6 carbon with a 5 carbon. If it's 6 carbon with 5 carbon, what we call it? We call it a purine. Now, what are the examples of purine? Adenine, A, guanine, G. Okay, they could be also only have that 6 carbon. Then what we call it? Pyrimidine. Pyrimidine. What are they? Thymine and cytosine. How they connected with each other? A corresponded with T and G corresponded with C. How they connected? What is this bond's name? Hydrogen bonds. Okay, let's review chemical bonds. What are the three chemical bonds? What we talk about in the animal or plant science area? Hydrogen bonds, ionic bonds, and covalent bonds. Can you give some of the example? What is covalent bonds? You had a water molecule. How they connected here? This is covalent bond. What is ionic bond? Calcium chloride. Arrangement of the electron. Hydrogen bonds right here. Hydrogen bonds is the weakest one. Ionic bonds, relatively weak. Covalent bond is strong. Now the covalent bonds, we also can separate them into polar and non-polar. Okay, what is polar one? H2O. What is non-polar? CH. Is that right? So, based on what? Electron negativity. Now, who created this? A chemistry name called Pauling. An American scientist. Wins the Nobel Prize 1954 and 1959. I want to mention this here because I want to use this again, okay? So we expand a little bit to talk about this. Electronegativity is also, Pauling talk about that. Okay, we talk about the base. How about here? What is important here? Always remember a phosphates. Is that right? A phosphates is important. Never forget there is a phosphates. Okay, this tri-polyphosphates is important because each individual nucleic acids, at this moment we still call it a nucleotide because it's not polarization yet. They each carry on these tri-polyphosphates. Then during the replication, they're going to hydrolyze them and connect it to the long chain. Now the question becomes how they connected them. How they connected. So we go here. Is that right? So you find another guy. And then they connected right here. This is from 5' to 3'. So they go this way. 5' to 3'. Generate a bond. What is that? What's the bond we call? 5', 3' diester bonds. What we are missing? Phosphate. Important, right there. Okay? So this is the whole process we talk about. Is DNA structure, basically. Remember Pauling here. Okay, then we're going to move on to another thing. 1952 we talk about. People know DNA a little bit. Then what is the structure? This comes out. The most famous and the single most beautiful scientific article in the 20th century is Crick and Watson double helix structure. Who is Crick? Who is Watson? James Watson. James Watson is an assistant professor from Indiana University, University of Indiana. He liked to watch birds, so he called himself birds watcher. In 1950s, he go to England as a visiting scholar, go to the Cavendish lab. Who is Crick? Frederick Crick. At the time, he's a physician. He had a little bit of experience about crystal. Their age is about 12-year difference. But they get along very well. So they went there, then they talk, they see something. It's a theory, remember, it's not an experiment. And they do different people, they talk every day. Now, how they get this idea? We know they use X-ray, the fraction methods. But I tell you one thing, before they jump out that paper, one of the chemists in the world from Columbia University, Shawgraf, have a lunch with them. What Shawgraf find? In any of a bacterial cell, it could be different from organism to organism. The ratio of A and T and G combined with C is always 1:1. And he had a lunch with them, he shared with the news to them. I tell you, when Shawgraf had a lunch with them, Shawgraf told all the reporters, they're not going to come out with this idea because they always talk and always think and never do anything. However, once they know the story, they come out with the double helix theory. They come out with the single most beautiful paper in the scientific area. When they find these results, what they do? They go to the pub of England, have a beer. They buy everybody a beer, $2. Everybody's happy. So they say that we had a breakthrough in the scientific area, nobody understands, but they enjoy the drinking. Then they go back, they write an artist. Or write an article. It's an artist, to be honest. And I tell you, when James Watson is a true scientist. He died in 2004 in California. About like 94 years old. You probably heard about news when you were in elementary school or in middle school. When I was at my senior year in the same college, he still reviews the manuscript. He's still revising the manuscript. So he's a true scientist, to be honest. So the topic here is, it's never single. The idea comes out by themselves. It's lots of people's work accumulated. And I tell you one thing, why I mention Pauling here. This is America's greatest chemist. He has so many theory about that. What is another most important theory is hybrid orbital of electron. So you don't like these chemicals. Look at the order. What they arrange? 1s2, 1s2, 2s4, 2s2, and 2p4. That's hybrid orbital of electron. It also come from Pauling. At the same time, Pauling also think about this way. Also think about the DNA structure. Could be some crystal, could be some helix. He is also getting there. In 1952, he published a small paper. Getting there. But at the end of the day, Watson and Crick Crick and Watson, they solved the problem. So I gotta want to tell you one thing. At that time, there is no biochemistry. There is only physics and chemistry person. They worked really hard to get in there. So that's why today you have a DNA, you know DNA structure. You can run a PCR in like 1 day or 2 days, get a fabulous paper. But all these people work really hard for all these material. Okay? Okay, before I'm gonna end this lecture, I want to mention one thing. What is really the implication from the double helix? Okay, we have this double helix right here. Okay, you know that. So beautiful. What's the implication? Semi-conservative DNA replication. So, in our next lecture, we're going to talk about detail the semi-conservative DNA replication. And we want to mention another important experiment before we talk about the detail and the enzyme. So thank you very much. I'll see you in another video.